everyone. Myself Sheikh Ismail Ali Basha, Department of Botany, Kevin College. Today discuss about third chapter Mendelian and non-Mendelian genetics. The Mendel's law of inheritance. The inheritance can be defined as the process of how a child receives genetic information from the parent. The whole process of heredity is dependent upon the inheritance, and it is the reason that the offsprings are similar to the parent. This simply means that due to the inheritance, the members of the same family possess similarly characteristics. It was only during the mid 19th century that people started to understand the inheritance in the proper way. This understanding of the inheritance was made possible by the scientist named as a Gregor Mendel, who formulated certain laws to understand the inheritance known as the Mendel's law of inheritance. This Mendel's law of inheritance, law of dominance, law of segregation, law of independent assessment. Next one is the codominance and incomplete dominance. The codominance is meaning two parent phenotypes are expressed together in the offspring. The incomplete dominance, two parents blend together to create a new phenotype for their offspring. Dominance, the two alleles neither act as the dominant or recessive over the other. One allele is not completely dominant over the other. Other characters, a hybrid will not result in the formation of the new phenotype. The incomplete dominance, a hybrid will always result in a new phenotype. Example, individual with blood group ABO exhibit the codominance. A and B are dominant in the relation to O. However, they are not dominant against each other. The incomplete dominance example, Snapdragon, Antirrhinium majus, the pink flowers are the result when the cross between the homozygous red flower and a homozygous white flower. Multiple alleles. The presence of the three or more alleles for a given gene is referred to as a multiple alleles. Different or opposing variation of the gene are known as alleles. Characteristics of a multiple alleles. The multiple allelism on the other hand occurs when more than two genes form the exhibit for the same locus. For example, one allele of the gene producing the height can be tall while other allele can be dwarf. It simply indicates that for a given gene, three or more alleles are present in the population. Only two genes can be inherited from each parent, be a human. Complementary genes. Complementary genes are a kind of gene interaction in which two genes interact to produce a specific phenotype or observable trait. A complementary gene is an interaction of two dominant non-interallelic genes in which each gene have its own effect, but when come together to interact, a new trait is developed and the Mendelian ratio 9:3:3:1 is changed to 9:7 due to the complementation of the both genes. This is the picture of the complementary genes, the cross F1 generations. Supplementary genes. The supplementary genes are genes that contain two non-allelic pairs of genes. They are both having an effect on the same characters. First gene is dominant and therefore can express on its own among the non-allelic genes. The second gene is also dominant, but it is only expressed when the first gene is present. Duplicate genes. The duplicate dominant involves the masking of two recessive alleles present at two different loci in the presence of a dominant gene. Complementary gene action is the alternate name of the duplicate dominant epistasis. When a cross is made between the triangular seeds dominant and the ovoid seeds are recessive. Due to this, 15 offsprings have a triangular seeds and one offspring has a oval seeds. Hence the ratio becomes the 15:1. This is the capital A, capital A, capital B, capital A triangular, and ovoid is a small a, small a, small b, small b. It is the common A, B and small a, small b. This triangular, the small a, capital A, small a, capital B, small b. Linkage and crossing over. The linkage, it is the tendency of genes and chromosome to remain and pass on to the offspring together. It is the breaking of the established linkage, the formation of new combination of parental genes. Linkage maintains parental combination of genes. It produces a new recombinants. It helps in the maintaining the newly improved variety in linkage. The crossing over, it helps the providing the raw material for the production of the new varieties. The strength of linkage increase, the distance between the two linked genes decrease. But crossing over, frequency of crossing over decrease with the decrease of a distance between the genes. The linkage become weaker with the increase the distance between the genes. The increases in distance between the genes increases the frequency of crossing over. Linkage increase with the age, crossing over occurrence the decrease with the age. The chromosome mapping. Chromosome mapping involves the identification of the location, structure and function of the every gene that is present on each of the chromosome present in the nucleus. The Human Genome Project is the wide successful genomic attempt by the scientists to map all the genes that are present in the human chromosomes. The 2-point test class. A test class is done between the dominant phenotype and a recessive phenotype to estimate the zygosity of the dominant phenotype. A 2-point test class is done to calculate the recombinance frequency of the two linked genes. It is useful for determining the frequency of gametes they produce. If there is a deviation from the classical Mendelian ratio, then we know that the two genes are linked and present on the same chromosome. 2-point test class example. 3-point test class. When the measuring the recombination between the three linked loci, the sum of the two internal recombinant frequency is the greater than recombinant frequency between the outside loci. The best estimates of the map distance are obtained from the sum of the distance calculated for the shorter sub-interval, which demonstrates a double crossover and shows only the middle gene being the altered in such cases versus the result with the single crossovers. A particularly efficient method of mapping three genes at once is 3-point cross, which allows the order the distance between the three potentiality linked genes to be determined in a single cross experiment.